First of all, thanks a lot, Leonard, for giving me the opportunity to present the work of my group and my collaborator at this workshop. Okay, so I think that if I'm saying that quantum technology are going to revolutionize many areas of photonic research, you agree. And fluorescent microscopy, and more in general optical microscopy, is not immune to that. So today what I want to show has been, as Leonel anticipated, I want to show our technology, a quantum technology, specifically this single photon avalanche diode detector array, or how we call them SPAD array during the talk, can somehow trigger to what I consider in the next generation of fluorescent laser scanning microscopy. So, okay. Laser scanning microscopy is probably one of the most used architecture, microscopy architecture in life science, together with wide field microscopy. The reason of that success is not only because it can be combined with fluorescence and thereby giving to your analysis molecular specificity and sensitivity, but also because it can combine with many other advanced microscopy techniques. So I'm talking about, for example, on confocal microscopy. So you know that by using a confocal microscopy, you can navigate across the third dimension of your sample. So you're not restricted only to make imaging on a layer. Or I'm thinking about two-photon excitation microscopy, so that can allow you to go deeper and deeper in your sample. Or fluorescent fluctuation spectroscopy. I'm sure that that's one of the great topic of this workshop, that give you the possibility to investigate very fast dynamic, or fluorescent lifetime, which open you to functional imaging. So anyway, at the end of the story, thanks to the combination of this technique, fluorescent laser scanning microscopy give you a unique combination of tunability, performance, spatial and temporal characteristic of your to your microscope. Specifically, give you this typically subcellular resolution, or even if you or when you combine, for example, on SPAD microscopy, as been shown before, you can achieve even below. You can achieve also subdiffraction resolution. Give you exceptional spatial range, so you can, as I told you before, you can image deeper, you can image a very large field of view, and so on. Also, great temporal resolution, depending by which kind of technique that you apply. If you use imaging, maybe you are a little bit slow, but if you combine with fluorescent fluctuation spectroscopy, you can observe very fast dynamic. And very, very important, and the end of the story is that fluorescent laser scanning microscopy does not provide you pretty nice image, but provide you really data. So it's a very high information context technique. When I established, yeah. So nevertheless, that's fluorescent laser scanning microscopy. It's a really powerful tool. When I established my lab at the Italian Institute of Technology, we came out with, we thought that anyway, this performance can be further improved because current laser scanning microscopy are affected by fundamental limitation. And to try to show you how we solve this limitation, first I have to introduce such a limitation. And to do that, I need to introduce a little bit how it works a laser scanning microscope. I know that Stephanie already did a little bit. I will recap really fast. So basically, in a laser scanning microscope, what you do, you take a laser beam and you focus by using an objective lens in a specific position of your sample. So you typically take this laser and you focus on a diffraction limited spot. All the molecules that are in this diffraction limited spot will emit light. So I'm talking about fluorescence, so will be excited, and then they will emit light. The light is collected by the same objective lenses, and by a combination of lenses and mirror and other optical devices. What happens is that an image of your excitation or probed region is obtained in the image plane of the microscope. So, and here typically what you have a single element detector such as a photomultiplier tube, that what is doing is integrating all the light that is coming to your image plane and that arrive to your image plane, and that is coming from your probed region. So basically you integrate all this light across the space. So nevertheless, you have an image here, because that's a microscope. You create an image in the image plane. What you have a single element detector that integrates all this light across the space. So you clearly see that there is something already wrong. So in principle, I have an image, but I'm integrated the light, and thereby I discard all this special information. Then typically what happens, I have a data acquisition system that what is doing is also integrating in time. So I mean, nevertheless, this is an image that changes in time, because obviously I have a molecule that may be diffused in and out from your probing region, or simply because you know that fluorescence is dynamic. It has a fluorescent lifetime behavior, so it has a dynamic behavior. But what I'm doing, I'm simply integrating light also across time. So I end up with a single intensity value. So again, I have some temporal information, and unfortunately, I discard this temporal information because my data acquisition system is integrated across the time. So then typically what I do, since in this case, I'm just making an example of an imaging system, what you do, you scan your probed region across your sample. Nevertheless, you end up with an image. You integrate all light across the space and across the time to generate a single intensity value, and you plot this intensity value in your matrix. You generate this raster scanning image. So anyway, what happened? At the end of the story, I hope that I was able to convince you that during this operation, a lot of information, a lot of single photon information, both in time and space, are lost. So how I can compensate for that? There are obviously solutions. And one solution is, for example, to stop using photomultiplier tube and using, for example, such single photon detectors like APD or SPAD, in which you can really sampling your fluorescence photon by photon at very high temporal resolution. But also in this case, what happened to the spatial information? Unfortunately, these are single element detector, and thereby you cancel out all the spatial information. So another approach is to use a camera, a pixelated camera, a CMOS or a CCD. In this case, you preserve the spatial information because you can make an image of your probed region, but unfortunately, this camera are really slow. So you cannot go, the scanning cannot be really fast. And on top of that, anyway, you are losing the very fast dynamic of your fluorescence. For example, you are losing the fluorescent lifetime information and so on. So it was clear at the time that laser scanning microscopy has a limitation, and unfortunately, technology was not able to compensate and overcome this limitation. So what we do, we approach the Polytechnic of Milan here in Italy, and we sit together to try to develop the first detector, and specifically detector array and the SPAD array, which is able to overcome all the limitations that I told you before. And we came out with this detector, and I will go, that from now I will call a SPAD array, a single photon avalanche diode array, which is the core of the Bright Eye project, that is an RC-founded project. And I will try to show you how with this detector now we can solve this limitation. First, I need to tell you a few details about this detector. I will not go too much into the detail. I can provide you the literature to do that, but I need to explain two very important features. First of all, that's a pixelated detector, but it's a very small detector. It's only 5 by 5 elements, so very, very few elements, not a megapixel camera like maybe you get used with it when you are using the CMOS. The reason of this very small size, because your probed region is very small. It's only a few 100 nanometer in size. So you don't really need a big camera to make imaging of a very tiny region. And 5 by 5 or 7 by 7 we are using is enough to sampling this image, satisfy the Nyquist criteria. And since we have so few pixels, the other very important thing is that we can transfer the data as fast as possible. So in the sense that we don't have a frame rate, so that's a camera without frame rate. Every time that a photon reaches one of these active element of the detector array, a digital signal is fired and then can be acquired by your data acquisition without typically any delay. So that's very important. There is no frame rate. So you can acquire a photon from each element at a frequency that is in the order of tens of megahertz, because you just have a small hold off every time that you collect a photon. Then the other very important property is that that's a single photon detector, and that's a time resolution of few hundred picoseconds. So when a photon arrives to a single element, is immediately transferred to your data acquisition with a resolution, with a temporal resolution, a temporal jitter of the order of 100 picoseconds. So fully compatible with the photophysics of any, let's say, of fluorescence. So these are the two most important properties of the detector, but just to give you an overview anyway, starting from the 2019, when we got the first detector, there was a lot of development to improve the efficiency, to improve the fill factor, to improve the speed of this detector. And I'm happy to tell you that now General Instrument is also start to commercialize such a detector. Okay, so once we got this detector, okay, once we got this detector, we have another problem, that is the fact that we need to record the data from this detector. As I told you, this detector is able to deliver you a digital signal every time that a photon is collecting from your sample, but we need to record this data, and a specific recorder from all the 25 elements of the SPAD array detector. So what we need, what is called a time tagging data acquisition system. So a data acquisition system that is recording with a precision of a few picoseconds, because I remember that the precision of the detector is 100 picoseconds, the photon coming from this from your SPAD array. But not only, you need also to record many different synchronization signal from your microscope. For example, since this is a laser scanning microscope, obviously you want to record also when there is a movement of your probed region across the sample. And then also many other synchronization signals. For example, if you use a laser, a pulse laser, if you like to also record the timing on which your sample is being excited. So anyway, at the end of the story, my microscope is not anymore generating, let's say, images, but is generating a list of events, and specifically a list of events that can be a photon or can be a synchronization signal. With this list of events, also what we do, we reconstruct again a different data set that is a list of photons. So that's why we start to call this microscope the single photon microscope, because when you sit in front of this microscope, you do not collect any more image, but you collect a list of photons. And each photon is coming with some signature, some very important spatial and temporal signature. For example, in terms of spatial signature, what we know the position from the sample where the photon comes from, the position on which has been recorded into the image plane, and in terms of temporal signature, we know, for example, the delay from the beginning of the experiment, the delay from the shift of my beam across the sample, or the delay between of the photon the delay between the photon and the laser that generate such a photon. So anyway, at the end of the story, what we have a list of photons with a series of spatial and temporal signature, which are typically discarded by a conventional microscope. So once we have this single photon data set or photon resolved data set, what we can do, obviously, we can always reconstruct a conventional data set. We can always reconstruct a conventional intensity-based images by integrating, like was doing my detector before, or making some histogram. So anyway, I can always come back to the conventional representation of the data. But obviously, if I have a conventional representation of data, I cannot restore the single photon data set. So that's a new way to collect the data, so which is fully compatible with your previous way. But on the other side, it has much, much more information, much more spatial and temporal information provided by yourself. So thanks to this technology, what we do, we immediately introduce in our custom microscope, laser scanning microscope, and we saw that we can in principle increase the performance of, increase most of the performance of your laser scanning microscope. So for question of time, I will just present only a few of them. And I will start with one example in which we use this information, and specifically we use only the spatial information provided by the detector array to improve the resolution of your laser scanning microscope. So now that we introduce this SPAD array into the microscope, so what we have, we do not have anymore a conventional image, but what we end up is that, as I told you before, for each probed region of my sample during the scanning, I collect what is called a microimages, as you can see here on the right. Or on the other side, what you can see, or you can see the same data set also as a collection of 25 different scanned images, as you can see from this part. So if you consider your element, every element of the detector as a single element of your detector array as a single element detector, basically every detector will generate an image that is more or less the same, or anyway represent the same sample for sure, but collected from a slightly different point of view. So basically any of your 25 images that you generate during the scanning process has a slightly different points per function. So, and as you know, the points per function is a way how you characterize your image. And in particular, you have 25 different points per function, which are very similar in shape, but they change in position. So what does it mean? That when I collect my 25 images, they have a different points per function because you can see them from a slightly different point of view, but all of them are similar. So what you can do, you can reconstruct a super resolved images by simply, what you can do, you can simply take the 25 images, register each other, because these 25 images are the very same sample, but shifted in space. And when after you shift back all the images, you just sum them up and you obtain an high resolution images. And specifically, you obtain an image that has a doubling resolution respect to a conventional microscope. And very important, that's, I can skip this slide, very important, you collect also most of the light from your sample. So basically, as you know, typically in a confocal microscope, you can also achieve high resolution, but you need to close the pinhole and thereby reject it off a lot of light. With such a system that is called image scanning microscope, you collect all the light because your detectors are in parallel collecting signal, and then every element is acting as a pinhole, and then you can, by computationally, reconstruct an image with high signal to noise ratio and with high spatial resolution. Here is an example, is a tubulin example in which you have you make imaging in a confocal modality with a very small pinhole, 0.2 area unit. As you can see, you can barely distinguish the structure in your sample because the image is very noisy because I closed the pinhole. So if I open the pinhole, what happened of the microscope, what happened is that I increase the intensity because I collect all the light, but unfortunately, I also decrease the resolution of the microscope. What I can do if I apply image scanning microscopy, and specifically, if I collect these 25 images and then I reconstruct them by pixel assignment, I will end up with a very high signal to noise ratio images and also with the same resolution of a confocal microscope. So as you can see here by this intensity profile, you can clearly distinguish two different filaments close by that you cannot distinguish if you open the pinhole, the green light, or if you close the pinhole because your image is too noisy. We are a group of computer scientists, and thereby we apply also much more convoluted reconstruction algorithms, such as, for example, a multi-deconvolution algorithm. So as you can see here, you further improve the contrast of your images. Again, what is very, very important here is that you don't have to change anything basically in your sample or in your setup except the detector. So you just substitute the single element detector, the photomultiplier from your confocal microscope, you add this SPAD array, and you end up with a system that provides you doubling resolution respect to your confocal microscope. You can use in any kind of sample, not obviously tubulin like I showed you before. Here we use for to study the chromatin organization inside the nucleus. It's clearly how the resolution improve when you apply image scanning microscopy. We can use also to study neuron spine. Again, here you can see how much the resolution improve. And again, without any change in your sample preparation or without the need of using a high laser like instead microscopy. We can combine with two-photon microscopy, obviously, because two-photon microscopy basically is a laser scanning microscope in which you use a nonlinear regime for the excitation. Again, you can clearly see the advantage in resolution using the SPAD array instead of a conventional photomultiplier tube. Another important aspect in the concept of deep imaging is that the micro images, so the image of your probing region, is also able to tell you if your light is coming from the focus or if your light is coming from out of focus. So without to go too much into detail, I can classify, thanks to the spatial information provided by the detector array, out of focus light and in focus light, and specifically I can separate them. So here is an example of a three-dimensional scanning. The left side, this image on the left, is a conventional images. The one on the right is an image in which, thanks to the SPAD array, we was able to separate the background, the autofocus background, and thereby improving the contrast of your imaging. So again, that's for free, completely for free, just changing the detector array, that's changing the detector of your microscope. So I want to move, how much time I have? I still have some time.
Yes.
What? How much time I have?
20 minutes.
20 minutes. Okay, great. So yeah, I can touch other two important projects that are currently running in our lab. So as up to now, I just show you how we can take advantage of the extra spatial information provided by the detector array. But as I told you, this detector array has in principle a limited frame rate and can achieve the single photon information with a temporal resolution of 100 picosecond. So that means that it's fully compatible with the fluorescent lifetime. So I can, in this case, what we do, we change the laser of our microscope and we introduce a pulse laser into the system, and and we make a time-correlated single photon counting measurement with all the 25 elements of the SPAD array. The only limitation that we had on succeeding this project was the fact that when we start the project, unfortunately, was not possible, or anyway, was not economical, affordable, having a card that is able to collect simultaneously 25 and in the new version 49 channel from our detector array. So what we do, we develop our time tagging platform, our time correlated single photon counting board, and we use an FPGA. So we develop 25 and in the current version 49 time to digital converter directly inside to the FPGA of our data acquisition system. And I don't want to go too much into the detail of this technology, but anyway, this technology provides you 30 picosecond resolution, so exactly what we need for your lifetime measurement. And what is very important, that's an open source project. So in principle, and I'm happy to support you, you can go on our website and download all the code and all the part lists to build up your time correlated single photon counting, not only for the SPAD array, but anyway for any other single photon detector. Now that we have this card, we can combine image scanning microscope with the lifetime information. So that's again an example of imaging of tubulin in which you can appreciate the improvement of resolution when you use the spatial information provided by the detector array. Now we can overlap also the fluorescent lifetime information. So now we end up with a fluorescent lifetime map that is double in terms of resolution respect to a conventional microscope, because we can take advantage both of the spatial and temporal information provided by the detector array. That's not really a fancy example because it's tubulin, the lifetime is constant, so there are nothing to learn from the functional point of view. Here is another example in which we use a polarity sensitive membrane dye, the ANEP, which is able to indicate the order of the lipid in your membrane. As you can see, in the plasma membrane, you have a lifetime that is longer, because it depicted an ordered phase of your lipid, while in the inner vesicle, you have a lifetime into the blue. That means that is shorter because you have a much chaotic situation. So again, you have a microscope. Obviously, that's a lifetime, that's a lifestyle image in which you combine a fluorescent lifetime and super resolution, at least doubling the resolution of a confocal microscope. Another example that we are running in our lab is to use the lifetime to understanding if a GABA receptor anyway are open or closed by simply measuring the fluorescent lifetime of a GFP protein. Another example, another very important example that we are exploring in our lab, and which try to leverage the new information, especially temporal information provided by the SPAD array, is in the context of fluorescent fluctuation spectroscopy. So I think that you are aware that if you are using a laser scanning microscope, since you need to do raster scanning, unfortunately your temporal resolution is limited by how fast you can build up your images. So thereby some specific dynamics that are in your sample that are faster than the frame rate of your microscope cannot be really investigated. But if you combine with fluorescent fluctuation spectroscopy, you can get access to study dynamics that are faster than any other imaging technique. So just to recap a little bit how fluorescent fluctuation spectroscopy work, but probably you already saw really a lot of other talk about that. So anyway, what you have, that's an image. Yeah, that's a video. So what you have now, you keep your beam stationary in a specific position of your sample, and you observe the molecule entering in and out the probe, the molecule or the particle that you want to investigate, the dynamic that you want to investigate, entering in and out of your probed region and generating some intensity fluctuation. So if you analyze this intensity fluctuation, and the most common way to analyze this intensity fluctuation is by calculating the autocorrelation curve of your intensity time traces, you end up with some information about the dynamics of your molecule. So for example, you end up with the transit time of your molecule. So how in average the molecules stay in your detection volume. And clearly this is a parameter that tells you a lot about how fast the molecule are diffusing, and thereby how fast the process that you are studying. You can get access also to other important information like the average particle number that you have in your probing region. So, but anyway, now we are mostly interested into understanding dynamics. So that's clearly a fundamental parameter. So longer is the transit time, lower is the molecule that are crossing your detection volume. And now that we have our SPAD array into the system, so we do not have only one single intensity time traces, but what we have is, in this case, is 25 different intensity time trace, and every intensity time trace is associated to a specific position into my detection volume. So what we can do, I can take this new data set, this new hyperdimensional data set. Basically, I have other two important spatial information that I can use for my analysis, and implemented many different methods. Some of them are well known, some are a little bit more novel. Again, for question of time, I just will present you the most straightforward way to analyze this data, which is called the spot variation FCS or diffusion law FCS. So basically now you have, for each element of your detector array, you have intensity time traces, and what you can do, you can sum up some of this curve in different subsets. For example, you can take the element, only the intensity trace from the the central element, or from the 3x3 element of the SPAD array, or the 5x5. And by doing that, what I'm doing, I'm mimicking different detection volume, and specifically detection value that increase from 140 nanometer up to 280 nanometer. And if I plot the transit time, or also called the diffusion time, as a function of this volume, I'm able also to distinguish which kind of modality I have in my sample. For example, this is the behavior that's the diffusion law. So again, diffusion time versus the size of my probed region. If this line is intersected at the 0, then you can say that your molecules are moving by free diffusion. If they intersect in a negative part, they are moving following a meshwork behavior, or if they intersect in the positive part, they are moving as they are, the molecule are affected by a modality that is characterized by some microdomain. So you have a sort of binding in some domain and then release. So that means that with a single measurement, we are not only able to tell you how fast the molecule is moving, but also which kind of modality the molecule is moving. Here is a phantom example in which we observe a molecule, a probe. This was Alexa 488 in PBS. As you can see, if we reconstruct the diffusion law, again, detection volume as a the transit time as a function of the detection volume. In this case, we see that the line intercepts the zero because obviously we expect a free diffusion from the molecule. More interesting if we move in a live cell, like in these cases here. So what we measure is a is a free GFP moving the cytoplasm. As expected, again, we can reconstruct the diffusion law, and the diffusion law indicates us that the molecule, the protein is diffusing free inside the cytoplasm. More interesting, when we go to start something biologically relevant. For example, in this case, we go to investigate a DEC protein inside the cytoplasm and inside the nucleus. As you can see inside the cytoplasm, if we plot the diffusion law, again, that's a single measurement. In a single shot, you can get such information. You can get the dynamics, how fast the molecule moves, but more important, you can get how the molecule is moving. In this case, again, free diffusion, but when you are inside to the nucleus, you can see that there is a completely different behavior because it's characterized by a crowd environment, and specifically here we have a macrodomain behavior. Another example that we are studying is in the context of stress granules, so in the formation of this kind of condensate. Here is an example, a measurement in which we measure G3BP1. This is a protein that is used to specifically target and specifically observe the stress granule into the cell. If we measure this protein before inducing a stress, before inducing the formation of some stress granule, as you can see, you have a meshwork behavior. After you induce the stress, adding some sodium arsenide, then the dynamics, the mobility of the protein completely changed, because now we have a completely different behavior of the diffusion law, which is better described by a microdomain modality of diffusion. Here is another example, just to compare the different modality of two different proteins, specifically the G3BP1 that I already showed you before, and another very important protein in the context of ALS, that is FUS, which is an RNA binding protein, which has been studied that the specific mutation of this of the gene that encodes such a protein is linked to the disease. So in this case, we do not show the diffusion law. We do not use the diffusion law as I showed you before, because instead of plotting the transit time as a function of the observation volume, we plot the diffusion coefficient as a function of the observation volume. Obviously, the diffusion coefficient is connected by the transit time by a linear relation. So more or less the data contained on this representation is the very same one. But in this case, free diffusion is characterized by a line that is straightforward, so does not change across the observation volume. Or when it's a meshwork, what we have a negative slope, and when there is trapping, we have a negative slope of the diffusion law. This graph here on the right side plot this diffusion law in a slightly different way, because what we do, we introduce this modality, this parameter that is called modality of mobility, which is simply the relation between the diffusion law of the smallest post respect to the diffusion, the diffusion coefficient respect to the biggest observation area. As you can see here, there is a complete change of the dynamic of the G3BP1 after and before the stress, as I showed you before. Interesting, in this case, there is a change of the dynamics, a change of the diffusion coefficient, and also a change of the mobility. In the case of FUS, having a conventional FCS measurement was not useful, because when you apply the stress, there is no change in the diffusion coefficient, but there is only a change in the modality of the diffusion. So that's, sorry, I forgot to tell you that each point of this scatter plot is a different measurement in a different cell, in a different position, in a different time. So anyway, in a single experiment, I can tell you which is the diffusion and which is the diffusion coefficient and which is the modality of diffusion. Obviously, I can combine with the lifetime, so correlating the diffusion modality with the lifetime of the molecule. And last, I want just to show really fast an example, another project which we are start to work quite recently, and is more into the direction of single particle tracking. So this topic has been already touched by Stephanie a few minutes ago. So what I want to tell you that basically now we have a detector array with a limited resolution, so no frame rate. So there is no any more limitation due to the detector array. And we can use this detector to make a single particle tracking a confocal microscope. So basically, as I told you, the detector array gives me an image of the probing region where, for example, there is a molecule or a particle. And since I have an image, I can also decode which position is the molecule. So for example, such in these cases, I can simply calculate, in this case, I can calculate every 10 microseconds, the position of the molecule inside my probed volume, and by calculating a simple center of mass. And what is very important, that when the molecule is moving outside of the field of view, because again, this is a very small camera, it's able to cover only one area unit, roughly 600 nanometer in size. But when the molecule is moving outside of the field of view, what I can do, I can use an open loop, again, another FPGA, to recenter my detection volume. Indeed, what I have is, since I have a galvanometer into my system, and I have also a piezo that is able to move along the optical axis, I can recenter the position of my detection probe in order always to have the molecule into the center of my camera. So to do it 3D, I need to make a couple of tricks, because what I need to introduce an astigmatism into the detection in order to be able to distinguish when the molecule is above or below my focal plane. So, but again, typically this is quite easy to introduce in a confocal microscope. So that's, I want to stress, that's a conventional confocal microscope in which I simply substitute the photomultiplier with a detector array, and I introduce a cylindrical lens in order to have to introduce astigmatism and decode the axial position of my particle. Here is an example in which we induce a change of a particle. In this case, was a gold beads, using this elliptical trajectory. So we have a second piezo that is able to induce the trajectory of my particle. And as you can see here, I can follow the particle across a relatively large area and localize it with good precision, which obviously also in this case depend by the number of photons. But again, here the limitation is not anymore the camera, but are the actuator. So I can track really fast phenomena. For example, here we can track it to a speed of 10 micrometers per second. That's an example on an imposed trajectory. So these are fluorescent beam move in water, which we tune the viscosity by adding some glycerol. Very interesting, again, at the same time, I can measure the lifetime. But what is very important that to measure the lifetime, we implemented time digital TDC directly into the FPGA that is able to make tracking. So I don't have to perform two separate experiments. I don't have to analyze the data in a different way. So as a last example, and then I finish, here is an application in which we start to track a lysosome inside to a living cell. In this case, we can track obviously the time which can track a phenomena depends by the brightness of the particle. In this case, we can track up to different minutes because the lysosome was really bright. There is no, in principle, any limitation in the tracking range because that's a confocal system. So it's limited by the field of view of your microscope that typically is in the range of 100 by 100 by 100 micrometer. And again, at the same time, I can measure the lifetime. So I think that I was a little bit long, so I will go really fast. I hope that I was able to convince you that if you add a SPAD array detector in your laser scanning microscope, you can improve almost any performance of your technique that you combine with laser scanning microscope. That's the tip of the iceberg. Much more projects are running in the lab, and many other people are following. And nowadays they can add this detector in them laser scanning microscope. Thereby, I really hope that in the future many new ideas will come out. All what I show you is part of a large open source project, which is not only delivering you the card to make the time tagging image, the time tag data acquisition, but also to build up your microscope and the software to control your microscope. The most important slide is the acknowledgement.